This is an interest of our group to look at drug resistance, and I think it's a very broad question. There are many mechanisms, and they are specific to each and every class and drug. But today we are revealing more and more of them. So it could be anything from down-regulating the target. For example, if we're talking about a monoclonal antibody, and then there is a downregulation of the expression of the antigen that this antibody needs to connect to. So like downregulation of CD38 could cause a resistance to DARA. We saw that with BCMA, sometimes there's even a deletion of the gene or down expression of the BCMA from the surface. Or there is sometimes cells develop, a way to overcome the pathway that is being inhibited by a different signaling pathway that is up regulated. So it's really a fascinating field, and of course very important for us to understand, so that we can perhaps find further targets and find mechanism where we can overcome these resistant mechanisms. As I mentioned before, we are actually using single-cell RNA-seq to try and find signatures of RNA that will predict various mechanisms of resistance. So first, this could guide us towards the selection of treatment, and also perhaps find ways to overcome resistance. So for example, we discovered a novel mechanism of resistance to proteasome inhibitors, which is to do with the overexpression of a gene called PPIA, which is involved in a unfolded protein response, and this could confer actually a resistance towards proteasome inhibitors. And we found that if we down regulate this gene, and we showed this in the lab by knockdown in cell lines, or even by inhibiting it with a small molecule with cyclosporin, we showed that we could actually resensitize cells to carfilzomib. So this is actually something we are currently exploring in a clinical trial as well.